This is the second part of the PDB inspired session. This is to celebrate the 50 years of the PDB. This is an absolutely invaluable resource that anybody who's involved in structure-based design will have used many, many times. So we're really looking forward to this. As usual, we'll be having a range of speakers. We will have a break on the hour for 10 minutes to allow people to get up and stretch their legs. And if you have any questions at any time, please use the Q&A box, and we will be monitoring that and transferring your questions to the speaker. We also have a panel discussion at the end, in which we'll talk about PDB in the future and things like that. And if you have any questions you want to put to that, then please feel free to. So without any further introduction, I'd like to hand over to the people who are going to be running the show today and ask them to take it away.
Thank you very much, Chris. Welcome everyone to the day 2 of this webinar. Let me just so I'm Samir Velankar from PDBE, and I want to thank Chris and Royal Society of Chemistry for organizing this, and also our colleagues from RCSB for putting the program together with us and for all the preparations. So today we are going to look at how to assess the quality of PDB structures and also the mol star viewer. And then we will take a short break, and in the second half we will look at the future of PDB, and then we'll have a roundtable discussion. So I don't want to take too much time, and let me hand over to John, who will talk about validation, assessing the quality of PDB structures. John?
Yeah, thank you, Smith. You should be able to see my presentation, I hope.
Yeah, we can see it.
Excellent. So hello, my name is John Berrisford. I work for PDBE, which is part of the wwPDB, and here I'll be presenting what we do as part of the wwPDB for validation of structures. So I'll go through several sections in this talk. I say, why do we need validation? I'll show you a bit of a validation report and some features within the validation report. And later on, other people will go through tutorials of how to use them as well. So why do we need validation? You can assume that all the structures in the PDB are perfect and that they have no errors, and they are great for all of your downstream analysis, but this is not the case. You may be surprised to know that we do not reject any structure based on quality. And because of this validation report, we now do, though, give an indication of the quality of the model that we have in the archive. All of the structures that we have in the PDB are models explaining the data. And this lovely quote here I like here, essentially, all models are wrong, but some are useful. I think is a fair representation of what we have here. So as I said, the PDB model is just an interpretation of the data. So for an X-ray structure that I have here, this is the data that we have. So you look at the electron density maps, and this is our model that we have in there. And you can see, hopefully, around here, if I, around here, this tryptophan, this phenylalanine, sorry, is in the wrong place. And it should be up here instead. And this is incorrect. But you would not know this if you did not look at the data that you have here. Not all structures that we have here are equal either. So for several of the techniques, electron microscopy and electron and X-ray structures, we have resolution as a metric. And actually this tells us the level of detail we expect within the structure. And just to give you some rough idea of what this means here for a ring, if you look at 3 angstroms, as we see here, we have a blob. Basically, as we move to 2 angstroms, we start to get some definition around this ring here. And as we move to 1.2, and even better if you go higher, you can start to see where the individual atoms are within this structure. And there's obviously some uncertainty at the lower resolution as to where the atoms should be. So what have we done as part of the wwPDB to address this issue and make users aware of what is in the archive? We have introduced a few years ago the wwPDB validation report, and all entries in the archive have a validation report. Near enough, there's a couple that don't, but the vast majority do. And these provide a consistent archive-wide validation view. What you have in the report is a PDF, which is freely available to download. We have validation data in XML and now in MMSIF format that's available for download and use programmatically. And we have some sliders in PNG and SVG format, which are used on our website, as I'll show you, and some external software uses them as well to tell you how good structures are at a glance. The sliders particularly are used for this at a glance view, and they really do highlight what's wrong. So what's in our report? We analyze geometry issues. So this is looking at the polymer, the ligand, chirality, all sorts of geometry. I'll go in a bit more detail. We analyze the ligands with geometry and again fit of model to data as well. And the whole structure is analyzed for fit of model to data, both in X-ray and EM, and slightly differently, but still true for NMR. How do we do this then? So we have a series of steps that we run for the different methods. And simply, we analyze polymer geometry, we analyze data quality, we look at the fit of the model to the data, we look at ligand geometry, and we compare structures to other PDB entries so you can see how good an entry is compared to other structures. And the software that we use varies depending on each method. In all cases, we use Molprobity for analyzing the polymer geometry. For X-ray, we use XTriage, EDS for analyzing the data, and then Mogul for ligand geometry. And this Mogul step, again, is common to all methodologies. For EM, the data quality and the fit of model to data differs. So we use something called vision analysis and volume inclusion. And I will show examples of these as well. And for NMR, this middle section is changed into chemical shift analysis and restraint analysis to compare the fit of the data to the model. So one thing I mentioned before is this slider. This gives an overview of what is in the structure. So it's a series of metrics that we have here comparing your structure to those in the archive and those with similar resolution in the case of X-ray. And it has several points to it. A metric that says how well does the model back predict the data for X-ray. This is measured in R3. We have clash score, which is how many atoms are bumping into each other. We have Ramachandran and sidechain analysis for looking at surprising bond angles, bond lengths. And then we have analysis of how well the atoms fit into the data. So slightly different from the top metric. Within the sliders here, we have, it's most obvious here, a black bar filled in and a sort of open white bar. The black one is relative to all structures, and the open white box is compared to similar resolution. And for obviously a good structure, we would expect everything to be up in the blue end, and for a less than ideal or structure warranting more analysis, we would expect things to be in the red end. So when we're looking at geometry, what are we looking at? As I mentioned, we're looking at bond lengths, bond angles, we're looking at clashes between atoms, We're looking at outliers within the Ramachandran plot, which I hope some of you are aware of. So do things fit into the expected regions that we have here? We check whether things have the right chirality, and this includes analysis for ligands. We look for the conformation of side chains and nucleotide sweetness, so a geometry measurement for nucleotides. For small molecules, for ligands that we have, we use Mogul to do the analysis. And we compare the bond angles and bond lengths that we have for parts of the ligand structure to structures in the CCDC database, the CSD. And we get obviously data from the CSD as to where we expect a bond length to be, and we compare that to what we have. Now, I wonder if you're thinking, oh, how necessary is this? Do we really need this? Well, we have some interesting examples in the archive to show just how much we need this and why this is important. So we have this lovely one here. I don't know how many you expect for the tryptophan, but this thing should be flat, rather than what we have here, which is what we call lovingly our toilet seat tryptophan, because of its shape. It's not expected, and obviously we highlight this in the report. And as you can see from these sliders, this isn't the only thing wrong with this structure. We have another few examples, some skeletons in our closets. So as it's that time of year, we like to bring out our models like this. So we have example of ligands that look like this. An interesting place to keep your hydrogens within the center of the ring. It's very neat, very tidy. Wrong, but that's how it goes. Ligands here where we can't tell by eye looking at where the connectivity should be between the atoms. So there's something wrong with the bond lengths and bond angles here. And then looking at fit of model to data, these are the same example, just rotated around. And here this looks quite nice. I mean, it's a little bit poor, the density, but it's not too bad. If we flip it round 90 degrees, what we see is that a large section of this ligand has no evidence whatsoever in the data. And these are the kind of things we're highlighting. So how do we highlight these things in the report? What do we do? So the first thing we do for the geometry analysis is we present issues on a sequence, just on a linear sequence. And we present it in two ways. We present it in a summary, which is shown here, and we present it in a per residue slider. In both, the color schemes and what we have is the same. So in the top slider, which is here is the 3%, which may not be present if it's 0, which is the fit of data issue. So anything that doesn't fit the data gets highlighted here. And in the bottom slider, we have various different colors depending on how many geometric outliers each residue has. So gray, there's a 12% here not modeled, 79%, so there's no issues with all of this. And then we move to yellow, where there's one issue on that residue, orange, where there's 2 issues on the residue, and red, where there's 3 or more issues, as you can see here in a small section here. And we take this same view and we expand it so you can actually see which residues have the problems. So if we look at that same chain that we have here, we can see that the N-terminus is all unobserved. And what we do is we collapse, every time there's a green bar, that's a large number of residues that we've collapsed into a bar when there's nothing wrong. So rather than displaying the whole sequence, we can shorten and only highlight the bad bits. And we can show with a red dot when it doesn't fit the data. And then we highlight each residue with its color as to how many issues it has based on the color scheme I mentioned before. We then provide detailed reports actually listing what are the issues with each one. So we can say which residues specifically are Ramachandran outliers here. And we have a detailed section of all of the different types of analysis for geometry. As we then move on to ligands, we treat carbohydrates as a polymer and sort of as a ligand at the same time. So we have a detailed report of all the bond length and bond angle issues within carbohydrates, and we draw a diagram shown here where we highlight whether there are issues. And as you can see with the magenta coloring scheme, this is where there are issues within this ligands, so you can see at a quick glance where there are problems. We do exactly the same for small molecules, so these two sections look very similar, and this is intentional, so that once you've learned how to view one, you can view another. But another section we add for ligands, which is important, is how well does the ligand fit the density? And we specifically drag out ligands here to show how well or poorly they fit. And this is a carbohydrate chain that we have in this structure, and I haven't cropped the map here. This is real. This is how it's deposited. This is the density around here for the protein parts, and that is the density for that ligand. It doesn't exist. It's not unique to specific cases like this. This has had some major structures have this. For example, this particular structure that we have here is a high resolution structure which is supposed to have diclofenac in it, and it is not supported by the electron density. And as of the time I wrote this, this was the only example of this drug bound, and it provides no evidence that this drug binds to this protein here. How do we highlight this in the report? Well, we show you a diagram where we plot the density on top of the ligand, so you can see at a glance whether it does or not fit, and we highlight it in a table whether the RSCC and RSR metrics for the ligand tell us that there is a problem. So that was X-ray. So I'll just go through some of the other two techniques of EM and NMR. So for EM, we do something very similar to what we did for X-ray. We highlight residues here which are not supported by the EM volume. Just because it's a slightly different metric that we're using, we highlight that it with a red diamond rather than a circle, just to say it doesn't fit. And we also present several images within the report to show a map model overlay, shown here, a plot showing the atom inclusion based on the recommended contour level, which is part of the entry, and then a series of images of the EM volume so that experts can judge whether or not there are issues with this representation. Similarly for NMR, we provide the completeness of the chemical shift assignments. We present statistically unusual shifts and something called the random coil endings. So parts of the structures that are disordered, and these are highlighted in cyan when they don't have a defined region. And I'll show an example of that. So here along that similar plot that we have of the sequence, we can see we have a new color, which is cyan. And that is, for example, these wiggly bits here, where they are undefined within the core of the structure. So they aren't similar between all the different models of the NMR entry that are deposited at the same time. So hopefully that has shown in all the ways that we highlight bad things. What I've tried not to say is that things are wrong. What the report does highlight is outliers. And these might just be things that are interesting, and they might be things that are wrong. And that's up to the user to judge and work through. For example, in this particular structure, this Val 48, which is highlighted just here in yellow, which is this residue just here, is supported by the electron density around it. However, it is an outlier. It is in yellow. It is a geometric outlier. It has a poor fit to the Ramachandran plot, but this is supported by the data in this particular case. So this is careful to judge structures and look at the things that are outliers and not just assume they are wrong. Very occasionally, we do get people who will redefine their structures based on what we have in the report. So in this particular example, this old structure had a very, very bad clash score. So all these regions are in red, plotted here are regions that have clashes with other atoms. And then on the right-hand side is the re-refined structure as redeposited as a different PDB entry. And as you can see just by eye, they are significantly different. And actually, you've got rid of all of the clashes within the structure. The one bit I will leave you with is, where do we show this data? So for people who are depositing structures, they have to review and accept a validation report before they can deposit. So they get to see this slider and all of the issues highlighted in the report before they deposit and after curation of the PDB entries. They have to accept it twice. We then present this data on the wwPDB websites. So here in, it's actually in light mole, but similar to the MolStar viewer that will be highlighted in the next presentation. So we can see all the different colors which match to here. And this is available in viewers sort of in MolStar and in external viewers such as Qt. Also, on all of our entry pages, there's the 3 members of the wwPDB who present PDB data. So that's PDB, RCSB, PDBJ. All 3 sites present the sliders on the entry pages so that they're readily available and you can easily see which structures have issues or potential issues. Okay, so just to conclude, the wwPDB validation report is a standardized validation for all entries in the PDB. This at a glance slider provide a very quick way of judging structures to see whether there's a problem or not, or potential problem. This is provided to depositors at the time of deposition, and the results are shown on all of the wwPDB websites for each entry. And just to acknowledge all of the many funders and the group of PDBE, which I'm part of, some of which you will see during this work. And thank you very much for your attention.
Thank you, John. Are there any immediate questions? Otherwise, I think we'll move on to the tutorial, which will explain this in more detail.
Yeah. Maybe
the
I could ask a
QA
quick
is empty at this point.
Yeah. Maybe I could ask a quick question. The resolution, I presume, is a measure of the entire structure. Can you get an idea of the resolution, say, around the active site?
We don't know. We don't provide that any analysis of that within the report as it stands now. Within X-ray structures, we do have B factor as the closest we can get. You can tell you how much movement we expect in a particular region. So regions with higher B factor are down as more mobile. And this is sort of, not quite, but sort of what you see in an NMR structure where you see more mobile, more flexible regions. But that's not really answering your question.
Yeah. Okay, thanks.
But I would just add, what's important really is that everyone looks at the RSR and RSCC values to see how good the side chain in the active site, for example, match the experimental data. And you then have at least some kind of measure to see if the site, the active site is good enough and explains the experimental data well.
There's a couple of questions for you in the Q&A now.
The first one's on color blindness. I'm afraid not in this particular case. The sliders, the reason that we have a red-blue slider is for exactly that reason. So that the most standard color blindness of red-green, you can actually differentiate between the two. And the diagrams I showed with the ligands, the reason that the color scheme of those is picked is because of color blindness as well. But when it comes to highlighting residues in the sequence plot, I'm afraid not. The only thing I can say is we do provide the data in a programmatic form that can be read and easily viewed.
So, John, the second question is about how to select a structure if you have many structures in the PDB for a given target.
Yes, it is something that we've had to think about quite carefully. Each wwPDB site has a different approach as to how to do this. The validation report itself doesn't offer anything. Obviously, it provides metrics of how good a structure is. At PDBE, we provide the results on search, so that with a slider, and they're sorted by quality. So you can see the top one would normally be the best one. I know at RCSB they do something similar with Ligands, but I don't think that's available in the search, where you can pick on the best instance of a ligand based on the validation reports. A question of, could you share the report? Does it mean, are they freely available to share? Is that what you're asking? If so, they're publicly available and meant to be shared with reviewers and journals at the time of submission to journals, and are freely available to share, as all PDB data is openly available.
I think we will look at some of those in the tutorial. So maybe it's time to move to tutorial and go through some of this.
Sure, thank you, John. Okay, so I'll move on to sharing my screen for the tutorial session. So what we've done is we put together, let me share the tutorial with you first. We've put together a tutorial that kind of walks you through these validation reports, reiterating some of the things that John's already gone over, but maybe giving you some examples of potential outliers, potential issues there might be with PDB entries that you can spot using the validation report, just to give you an idea of the type of information that you can get. So I'll share a slide in a minute with the link to the tutorial, but before I do, I'll just give you a bit of a run through, just to give you an idea of what the tutorial covers. So the first thing to say is that I've provided a link here to a folder where you can actually find all of the validation reports that have been mentioned in this tutorial. That said, in the tutorial, we've tried to provide you the information that you need for the tutorial questions as well. So you don't necessarily need them, but if you want to look at more comprehensive reports for those, that's where you can find them. So the first section of this really is just a bit of a reiteration of some of the things that John's gone over about the sliders, and reiterating how these assess quality and allow you to get an idea of the quality of the structures you're looking at. And again, this at a molecular level as well, how to assess the quality of each of the molecules within that structure. Then there's a bit of a run through here with individual outliers. So looking at individual amino acids and using the information in the validation report to determine why these outliers exist and how real they are. And as we go through, there's a number of questions we're just asking, just to give you some things to address. And at the bottom of the tutorial, all these questions are answered, so you can refer to those. We also have a section specifically on ligand validation. So in this case, we're looking at NADPH ligands and some of the information in the validation report to allow you to compare two particular entries to determine which of these ligands maybe is more reliable if you were using this data separately. And these use some of the images that John has mentioned that you find in the validation report. We then have a section where we're looking at specifically electron microscopy structures. So for these, we have data in the EMDB as well as PDB entries in the PDB, which are fitted to that map. And there's a section here which gives you an idea of how you can understand the quality of the fit of the model to that map, and how much you can rely upon the data there, and also a bit about the information related to contour levels and how this affects the validation of the entry. And then finally, there's a section just covering a bit like what was asked in the question about choosing the best structure for your needs, just a few ideas of things you will need to consider, and some example searches through both RCSB PDB and from PDBE as well, to give you an idea of how you might find particular structures that you're interested in and narrow it down to certain cases, and then also how you can sort these using those search pages. And as I said, in the tutorial answers we have down the bottom as well. So that's the tutorial we have. I'm just going to now share a slide which just addresses how you find those tutorials. So I've created a short link for these so that you can easily get to those. I will also add this to the chat so that we can, in fact, if one of my colleagues while I'm talking could add this to the chat so that people can access the tutorial pages. So it's thank you.
I have added it, David.
Sorry, Samir.
I have added links to the chat for everyone to go and look at those.
Great, thank you. So yes, this gives you a link here to the tutorial. Now we have around 10 minutes or so now until we're due to have James speaking about Molstar. So I'll just give you a little bit of time to have a look at these tutorials, to ask any questions you have through the Q&A about these tutorials. Hopefully everything should be there for you to take these away and look at them again in your own time, because we have the answers provided below. So we'll see what we can get through in the next 10 minutes or so, but we'll leave you to have a go at that for now. And then any questions, as I say, ask them through the Q&A and we can come back and answer those as we go. Okay, so perhaps we can go through one of these examples rather than we'll just wait and follow these. We'll go through one of these examples so that you can see how these work. Let me just stop sharing that so I can share the tutorial window. So I think one of the ones that John alluded to actually in his presentation was about assessing individual outliers. So in this case, we're presenting a molecule of cystatin A from PDB entry 3KSE. So this graphic here presents the residue level outliers as percentages related to each particular molecule in the PDB entry. So these are what we call our residue property plots. And it shows this for geometric outliers in the bar at the bottom. And it also shows, in this case, it's a very tiny percentage, but it would show any percentage of RSRZ outliers for crystallography entries. So that's residues with poor fit to the experimental data. So it then also shows this information at an individual residue level. So as we've put here, there are 11 residues with geometric outliers. So in these plots, we don't show, apart from the very first and individual residues between other outliers, we don't show stretches where there are no outliers. So you see here, we go from methylene 1 here to a glutamine, sorry, with an outlier at 17. So we kind of skip the ones which have no problems and aren't interested. So all these yellow ones here have single geometric outliers, and in total we have 11 of them. In addition to that, we also have one of these residues with a red dot above it, which indicates that it has an RSRZ outlier, so an issue with its fit to the data. So for this question, we're focusing on the valine 48 here and the aspartate 61. So both of these are yellow, so both of these have single geometric outliers. And just to cross-reference this and give you an idea based on the visualization, the image on the left will be familiar from John's slide. So this is that valine 48 shown in the Molstar viewer with the experimental data displayed as well. So one of the things we noted above is that this is a geometric outlier of some kind. However, you can see from the experimental data that it fits very well to that data. Whereas the one on the right, this aspartate 61, you can see that there's very little experimental data to back up the incorrect geometry that's been observed for this. So the question really we're asking here is which one of these outliers would you say actually it's a true outlier and you could trust this? Well, which one suggests it's probably incorrect modeling of the structure? So in this case, the valine we can trust because of the quality of the experimental data, whereas the aspartate, because there is no experimental data to verify this geometric outlier, most likely this is just incorrect modeling of the amino acid. So again, this is where John was saying how the fit of the model to the data is really important to verifying whether outliers are reasonable outliers or whether these are actually incorrect modeling. And the second question here is then relating this back to the validation report. So did this information in the validation report give you the information to determine this? Well, we kind of worked this out from the images, but actually if we go back to the residue property plots, where we noted that V48 was geometric outlier, but didn't have this red dot above it, whereas aspartate 61 did. So we could go back and say from these plots that it did give us an idea that this was a geometric outlier with a poor fit to data. And so you can take this information directly from the validation reports. However, if you're particularly interested in a region of a structure, then I think it is always good to be able to look at that electron density, look at the fit to the data, just to verify that is sensible and that there is sufficient data to back up the modeling that you're observing in that region. Okay, so we have just a couple of minutes before we start James's Molstar tutorial.
Can I another quick question then?
Yes.
So what happens if you have unnatural amino acids in the structure?
So we do validate. Let me get this right. We validate against the Ramachandran plot, and we do allow. There are allowed regions that are expected. So we will expect some outliers. However, we do have problems if there's a full peptide, which is D amino acids, for example, because they will, I think, regularly fall outside the Ramachandran plot and be flagged up as outliers. So these are particular cases where we would want to use that experimental data to confirm that these are reasonable outliers and these aren't errors in modeling. Yeah. I don't know whether, John, you have any other comments on this at all?
Hold on. Not really to add too much. The only thing we will struggle to compare them to other residues in the archive due to the number of them. So when you're looking at our plots of, say, this residue should be within the density, otherwise we'll flag it as a red outlier. we don't have enough of the instances of the non-standard residues to perform the same analysis for these. So that part is analysed slightly differently. But yeah, other than that, yeah, they cause us a few problems, and then again, so do ligands and all sorts of things that are different in different entries.
Okay.
And then that's the benefit of having visualization tools like Molstar, which I'll use as a way to bridge nicely onto the next talk, which helps to be able to then, for those cases where it's not easy to do it through the metrics, allows you the possibility to visualize it directly.
Yeah, a very nice segue.
In which case, at that point, I think it's perfect time to hand over to James.
Indeed, right. Can you see my slides okay?
Yes, we can.
Great, thank you, Dave. Okay, so good afternoon, everyone. My name is James Tolchard, and I'm a data curator at PDB Europe. During this part of the talk, we're going to look at the features and basic usage of the Molstar web tool for visualizing 3D molecular structures, along with their experimental data and annotated information. So to start with, I'll assume you're at least familiar with the principle of molecular visualization and are perhaps aware of a few existing tools already. Maybe the most well-known programs are the desktop-based ones, such as PyMol or Coot. These are the packages installed directly on your local computers, and they often have powerful features such as elements of refinement or docking, but lack full web integration. On the other hand, there are a number of web-based tools such as JSMol or NGL, and whilst these can be embedded online, they often have very specific functionality, and without the power of local hardware, they can struggle to display larger structures and volume data. So then with these different options available, you might actually ask, is there any point in developing yet another structure viewer? I would say, well, from our perspective, the answer is most certainly yes. The key point being that with these two approaches to visualization, there are large swathes of functionality that are mutually exclusive. Whilst there is, of course, room for a spectrum of solutions to the problem of visualization, we believe we can best serve our users and at the same time represent the complex data in the wwPDB archive by trying to bridge this gap between online resources and the functionality of local hardware. Therefore, it was with this concept in mind that Molstar was developed to provide a molecular viewer that was easy to use and suitable for embedding online content with, but which could also be used to explore aspects which are typically more computationally demanding. In addition, to remove some of the importance of local processing power and data transfer, we now also host specific data servers which are used to serve coordinate and volume data in a very precise manner, so as to be as efficient with data transfer, parsing, and processing as possible. Now, I'm pleased to say that the Molstar development has been and continues to be a collaborative effort. From the beginning, the core Molstar package has been co-developed between David Sennell of PDBE and CICAG and Alex Rose at the RCSB US Protein Data Bank, and new features continue to roll out on a regular basis. Now, in addition to the core functionality of Molstar, which you can find at molstar.org, its open source and modular code allows for the creation of unique instances of the viewer, which can be embedded as and when they're needed. You'll see a few examples today, especially on our sites, that our in-house developers have helped to implement. Now, at a rudimentary level, this can include customization of the general look and feel. However, functionality and performance can also be optimized by adding or removing different modules or functionality, or in fact by interfacing with other elements on a given page. Similarly, external data resources are now also starting to explore the potential of Molstar integration. One such example of this is Uniprot, which now uses Molstar to provide access to every related PDB structure on their individual pages. and the AlphaFold database, which uses MolStar for the presentation of their structures with their confidence score color scheme. And in terms of features, MolStar was designed to provide ready access to the most common visualization aspects like molecular representation and coloring, as well as overlays of biological assemblies or validation scores. There is, of course, the ability to pull structures directly in from the PDB with an accession code, and you can load local structures for visualization as well. There are also many advanced options to dig through, such as occlusion and other lighting effects. And thanks to the magic of WebGL, the images you create can be exported in high quality without taxing operations such as ray tracing. That's worth quickly noting that I've actually, hopefully it will work nicely, been able to embed MolStar directly into today's presentation using an iframe component on the slides.com website. Technically, I couldn't get this to work with our version of PowerPoint 365 or with Google Slides. However, as a free option, I thought it worth mentioning, as in the past I've spent a lot of time looking at ways to incorporate 3D data into my talks. MolStar has been developed with portable devices in mind, which includes options for different screen sizes, a full range of touch controls. And lastly, I will hopefully be able to show you in the next few slides some of the many ways in which you can access MolStar from our pages, including directly from search results. However, while this will be a bit of a whistle-stop showcase of MolStar, in case you'd like to try using it yourselves, you can also directly jump to a MolStar instance of your favorite protein by URL alone. And there are some example URLs at the bottom here, and you need only change out the 4-character PDB code highlighted in red. Okay. So for a live demonstration, we'll first look at the basic usage of MolStar and show you how to load a structure directly from an RCSB entry page. So when landing on a typical entry page, you will immediately have a set of 3D view links for the structure beneath the image of the biological assembly on the left-hand side. We'll look at some of these other possibilities later on, but for now, I'll just click on the structure link. Hopefully, everything works. Lovely. You should see an instance of the MolStar viewer on the page. So first and foremost, what you see here is the main MolStar section showing our molecule of interest. We then have a tools pane on the right-hand side here, and a quick access menu to the top right of the main viewer. In descending order, these icons allow you to reset the camera view, take a screenshot, toggle the controls pane on the right-hand side, expand to a full screen view, and then provide access to some more rendering options, and then finally toggle a selection mode as well. Now the mouse and touchscreen controls I previously mentioned work here in the viewer, and hopefully you can see the speed at which I can rotate around the structure, move it around the viewer, zoom into the protein, and then if I hold shift down, create a slabbed view as well. I'll just reset the camera now. There we go. Okay, with respect to the typical settings concerning representations, you can find these in the components section of the tools pane. Here, each molecule type or selection will have its own specific options for color and style, and the different components can be individually modified to your preference. Now, if we expand the options associated to the polymer visualization, you can see at the bottom here that the current style is in cartoon representation. And if we expand this option, we could perhaps change it to another type, such as ball and stick. And by simply clicking it from the list, we'll have it immediately applied to our selection. For colors, you can choose from many predefined color themes or uniform colors, or indeed other themes which reflect different biophysical or structural parameters. Now, all of these features, where applicable, also apply to ligands and water molecules, And you can access those representations in the same way and from the associated actions icon on the right-hand side. Now, you might have already spotted something, and that is also if you hover over certain areas of the protein, I'll try and keep my mouse steady, there we go. We're able to get quick access to the molecule name, chain name, residue type, and position for our selection. And you'll see in other sections of the talk how this information changes depending on whether we're projecting any other annotations onto the structure. And yes, also worth mentioning is that here at the top of the RCSB implementation of MolStar, you have access to the sequence view for the selected protein chain. And should you need it, you can use this to immediately zoom to certain parts of the protein in the 3D structure. Okay. MolStar is also able to help us look at and interpret volume data. In the case of X-ray crystallography, with MolStar open, we can find the density options in the basic settings pane on the lower right-hand side of the viewer. And this will appear for all X-ray entries. Now, if you're not familiar with these density maps, simply put, the 2FO-FC map should highlight the density into which the model was being built, and the emission map should highlight any extra density that either wasn't modeled into, shown here in green, or extra bits of model which are not well described by the data, shown here in red. So to look at that in practice, again, navigating from an RCSB entry page, this time we'll use the Electron Density Viewer link. This isn't strictly necessary, but ensures the density data will be immediately ready for us when we ask for it. Okay. Now, once loaded, we can see that by clicking on any part of the protein, it will automatically get a slabbed zoom to that residue, where the relevant section of the density map is fetched and overlaid on the structure. In fact, if I zoom out here a little bit, you can see really we have only retrieved a small section of this map. Now, this is an example of MolStar using the PDB volume servers and ensures we're only serving and you're only waiting for the data you want to see. Now, this is a quite particularly nice entry, and this is a good area. You can see there's a minimal amount of noise in the red and green emit maps here. However, should you want to investigate these further, there's also the facility on the right-hand side here to drop the visual threshold for any of these maps. And by clicking on the map names, you're also able to change the color and representation and opacity of the map as well. Now, lastly, should you want, you can also request the full density maps for the whole ASU. Hopefully now, if I do that by changing the view to whole structure and clicking update, you'll see how quickly we're able to retrieve the map, in fact, all three maps for the full ASU. Okay, moving on. As comparable volume data, the similar features also exist for visualizing EM map data in MolStar. Now, historically, visualizing EM data has required quite large data transfers. Other tools, for example, ask you to download the map files to your local machine. However, with MolStar, an EM entry with an associated map can be quickly visualized totally online. The first takes advantage of the volume server, as I previously mentioned for the electron density maps. However, we also improve the speed of map visualization for EM data by actively downsampling the maps when using a low zoom level. This is another method of us which helps us to be frugal with the data that's served to help reduce the potential of lag for the user. Now, in this example, we will open MolStar directly from an RCSB search result. So here you can see that I've searched for entries related to a particular PubMed ID. And we can see that this team's structure in this publication are a 20S proteasome and the tobacco mosaic virus capsid as determined by electron microscopy. Now, directly from the search result, I can launch MolStar by using the 3D view link here. Hopefully, there we go. And once opened, I can simply enable the density streaming clicking on density, and choosing to enable it. And there we have it, a full map fetched and visualized quite quickly. Now, similar to X-ray entries on the right-hand side here, we can see the density settings. For example, we can simply toggle the map on and off with the eye icon. And by clicking on the EM option, we have access to the color, representation, and opacity again for the map. Now, if we wanted to interrogate the agreement between the map and model here in specific detail, what I'll do for this presentation specifically, only for clarity really, is to change the view to around focus. And then under properties, we can change the detail level to a higher tier, 7 here, to get the best possible resolution. And then by clicking on any part of the protein, that's left clicking, will then be served up a high resolution section of the map. And for example here, we can start to see the helical density into which the model was built, and indeed also some sidechain information, or some sidechain density, I should say, into which this sidechain was placed for arginine 112 here. Okay. As I mentioned at the beginning, there is also the functionality to show annotations within the viewer, including those prepared by the PDB and other bioinformatics services. This includes examples of highlighting the difference between the structure in an ASU versus any biological quaternary structure or symmetry partners. Perhaps some of you are also familiar with the wwPDB validation reports, especially after John's talk earlier, and these are the ones we produced during deposition and alongside our public entries. Now, the simple color code that was described, ranging from green to red, describing the accumulation of geometric issues, can be used as a color scheme to help explore the quality of any PDB entry in 3D. And then we can also pull in annotations from other services such as PFAM to highlight extra features like protein domains. Okay, so very quickly I'll show you those in practice. This time using the iframe to pull a direct URL to a PDBE instance of Molstar for a structure of HSP90 with an inhibitor bound. So at the top of the control pane here, under type, we can see that the biological assembly is selected by default. And in this structure, that forms, or that assembly is comprised of 2 distinct protein chains. However, if I change type to show simply the model that was built in the deposited file, we can see that the dimer was constructed with a symmetry operation and that the deposited coordinates in the ASU only describe the monomer. Okay, if I go back to the annotations pane, I can quickly choose to show the validation report quality scores, from which we can see, perhaps unsurprisingly, our flexible regions and the boundaries of our secondary structure have some of the worst scores. And indeed, if I hover over specific residues, we're able to see a bit more information in the tooltip on the bottom right-hand side here. So for this residue, aspartic acid 156, we have issues with the RSRZ outliers, bad bond lengths, and planarity issues as well. So if I turn that color scheme off, I can now choose to show the annotations from the external services, whether it be PFAM, which is showing, in this case, our histidine kinase protein family domain. And use Interpro to highlight the different subdivisions between the functional families of HSP90. And then also the CATH, there we go, the CATH structural annotations, which describe, in this case, the same thing, which is the annotation for the whole heat shock protein topology. Okay, now the last thing I wanted to mention today is that Molstar is also a great platform for investigating ligands and PDB entries. Now, for example, when landing on a PDBE entry page where ligands are present, specific links will be available next to each chemical component. So when these are clicked on, hopefully we'll be taken to a bound molecule page, presented firstly with a 2D view of the ligand. various descriptions of the neighboring protein residues, and then an instance of MolStar showing the ligands in 3D in the electron density. Now, if multiple ligands are present of this type, you can choose to select them from the drop-down box at the top. This will zoom to different ligands in the structure automatically. Now, in this instance of MolStar, to save space, the right-hand control pane is hidden by default. However, by toggling the expanded view from the quick access menu, we can quickly maximize the viewer to take a closer look. And from here, we can also then show all the controls as they're available before if you want to start to make more complicated selections. And lastly, as I said, for X-ray entries like this one, you can see that the 2FO minus FC map and emission maps are shown by default in MolStar on these pages. And that's to help you readily inspect the data, which was supporting the construction of these ligands. Okay, so in conclusion, I hope I've been able to show that MolStar is a fast and simple online viewer with powerful tools for analyzing complex structures. By drawing upon the links in the PDB, I hope you can see how MolStar has the potential to readily allow the comparison of structural data to their associated experimental data, that it does so efficiently with particular regard to the visualization of volume maps. That being said, I'd of course like to acknowledge the wwPDB as a whole and their funding sources, as well as my fellow colleagues and curators here at PDBE, and specifically David Sennell and Alex Rose for their direct development of MolStar. Okay, thank you for staying to the end, I suppose. Hopefully it's not been too much of a blur. If so, there's extensive documentation for MolStar available at molstar.org, as well as some easy to find tutorials on YouTube as well. Thank you for listening.
Okay, thank you. Can I ask one quick question? Can you show non-bonded interactions like hydrogen bonds, electrostatic interactions, things like that?
Hydrogen bonds, I believe, is shown. Where are we? Electrostatic interactions will not be. I can't speak for the ligand view. Where are we? There. But no, I do not believe it's an option in MolStar. It will only be in the 2D viewer.
So just to add to that, there is in the most recent update of MolStar, there was an option have a representation of non-covalent interactions. So that if you are using the up-to-date viewer, which I think is now in the PDB website, if you click on a specific region of the structure, it will show the non-covalent interactions. So I think in this case, James, if you go to an entry page rather than the ligand page,
right.
One of the things we've done is we've not included this on the ligand pages because of the fact that these are calculated separately from our own interaction data that we calculate at PDBE. So we have a much more comprehensive set of interaction data we calculate at PDBE web pages, but there is a basic hydrogen bonding interactions added through MolStar as well. So those are possible, and we're kind of working now on trying to make them a little bit more comprehensive as well.
Okay, that'll be really good. I think now is the time for the break, isn't it?
Yes, so if we have a short 5 minute, well, if we come back about 10 past 4.
Okay. Yeah, that'd be good. Tammy, do you want to just say a few words?
Yep. Thanks to everyone who's contributed to this excellent workshop so far. I'm Tammy from Liverpool Chirochem. We are a chemical technology innovator on a mission to accelerate small molecule drug discovery through expanding access to 3D chemical space. Liverpool Chirochem, we're proud to sponsor this series of workshops as we understand how essential these open source tools are for the global research community. For those of you who don't know LCC, we specialize in using stereo-defined N heterocycles for use in DEL, FBLD, bifunctional linkers, and our 3Discovery virtual library. If you would like to find out more about how LCC could support your projects, please reach out to us through our website, liverpoolchirochem.com, or find us on LinkedIn. I hope you all enjoy the rest of the workshop. Thank you.
Thanks very much, Tammy.
Thank you.
So we reconvene at 10 past then.
Yeah, let me just start the. Okay, can you see my screen?
Yes. Yeah, all looks fine.
Okay, so let's start. So I'm going to talk about the impact of the recent developments as well as the impact of advances in structural biology and how we are planning to on the wwPDB side to cope with the new changes. So I want to start with the day when PDB started. So this is the announcement from 20th October in 1971. And as you all know, we are celebrating 50th year of PDB. And this was the first open access resource in biology and a single global archive of all experimentally determined structures. Now, why archive structures and why have them open access? So the first and foremost, I think, is the accessibility. So you have one place where you can get all the data. And once you have a archive, you can start thinking about uniform data representation and annotation. And we heard on the first day about MMSAFE and how that helps in standardizing the representation of structural biology data. But also, as is evident, that PDB is now around for 50 years. And so this data that all the different researchers have worked on can be kept, and all that investment of time and resources can be made available for generations to come by having an archive. Now, those are two reasons that you could have a archive, but more importantly, once you have all the different data in one place, you can start comparing the different structures in this case, or as we saw, we can start thinking about how do we assess the quality of structures, how do we select the best representative for any research, and integrate that with other resources. And so I think one of the advantages that structural biology has is from the very beginning, there was thought given to data archive and open access. And as a result, we don't just have PDB, which archives atomic structure data. We have EMDB, which archives EM volume maps, and also raw data from EM is archived in Empire, and other data resources archive raw data from diffraction experiments. And this archiving started, and open access started showing its impact from very early on. So in 1986, this is a famous paper from Chotia and Lesk, and we started understanding the fact that during evolution, protein structure is much better conserved than protein sequence. And there was a lot of software developed and to also do homology modeling. So this is just one example to show that even before PDB started, the first homology model was built by hand in 1969. But then there were many data resources, as I show here, developed for getting structures based on homology modeling methods. The other, I think, big outcome of having all this data open access was the realization that there is residues that co-evolve, and those residues that co-evolve are in contact with each other in the 3D structure. And what that meant was that if we could derive information on coevolution, we could turn it around and say those residues are in contact, and we can predict structure based on that information. And this is just a couple of papers where this was first published, and this has played a great role in the AI developments that have happened recently. So PDB continued to serve the structural biology community, and in 2003, the wwPDB was formed. And the main reasons behind having all this coordinated effort that is worldwide is to make sure that we have data standards, and we make sure that we get the depositions, we annotate them, and we have clean data that we can serve back to the community. And so not just the deposition and biocuration, but the remediation efforts that wwPDB partners undertake are extremely important in making use of this data for the broader community. And then what we saw from 2000 was this great growth, exponential growth in all sorts of public data. So sequencing allowed us to have a huge amount of genome data, which then was translated into protein sequences and proteome. And so UniProt now has about 220 million structures. And at the same time, because of structural genomics and other improvements that happened in structure determination, the PDB has grown to about 184,000 structures today. Although if we see how much of protein sequences from UniProt are represented in the PDB, it's very small. But I think the information content in these structures that we have in the PDB is so large that that's what helped this data from sequences and from structures helped us to make this great advance in structure prediction. And as we know now, there are two major programs or pipelines from DeepMind and David Baker's team that can predict structures for a single molecule, and now also for complexes. And they are really good in terms of their quality and match the experimental structures in most cases. Although there are issues in how we should use these structures is a topic that maybe we can discuss in a different webinar. So what's the impact of these developments? Now we have a situation where we have structure for almost every protein, or we will have in a couple of months, structure for almost every protein in UniProt. How is that going to impact our the structural biology? And also, how can we use this data in understanding the biological meaning of all this information? So what we have seen from various on Twitter, if you go to bioRxiv, there are now many publications. The availability of the models, the predicted models, has accelerated structural studies. So we are no longer limited in terms of having a structure. We can use the predicted model as another tool to get experimental structures, right from maybe improving construct design or using it as a starting model for structure determination, or when we have low resolution EM maps, we could take these models and fit in them and do time resolved studies, etc. So this is amazingly going to accelerate the structural biology and the understanding of the structures in their context as well. So what I mean by that is one of the limitations of the integrative hybrid methods was also having these models available. And until now, we were limited to having homology models, and now we are not. So we have now models for almost every protein, which means we can use different experimental techniques to really study large macromolecular complexes like the nuclear pore. And we just saw the paper from Jan Kozinski's group come out recently in Science, where they have solved the structure of nuclear pore, doubled the number of components for which we have atomic resolution model. And not just that, this might also result in use of a combination of sparse experimental data and predicted model to understand biology and create some testable hypotheses. So it might be something as simple as having the model alongside maybe chemical footprinting data or hydrogen deuterium exchange. But we are now going to start understanding the biology in 3D by combining all these different techniques and making use of all the different techniques to really understand the biological problems. So the first problem we want to address as wwPDB is, how do we capture all this data? How do we make sure that all these different experimental data that is being used to understand biology in the context of a 3D structure is captured in the appropriate archives, is made available? And so as wwPDB, we are working to create this structured data ecosystem. And that includes then not just the core archives like the PDB, EMDB, and BMRB, but also these other archives that take the experimental data or the different data resources that predict structures, and make sure that we work with all of them to develop the necessary data standards, to develop the necessary mechanisms to access the data. And so I'll just give one example of that, which is this 3D beacons. And the idea here was to work with the different data resources that have predicted models. So for example, Swiss model, of obviously AlphaFold-DB, Gremlin, Genome 3D, and Modbase, to create a mechanism by which users can access data from all these different archives in one place. So create a portal where once you send in a query, you get back information that provides you access to all the different models, including from PDB. And this was also to improve the coverage of sequence space. As I said, PDB has really small number of data in terms of sequence coverage, sequence space coverage, but that gets augmented by all these other data resources. Obviously, we have to make sure that it is quite clear to users that PDB is experimental data while the others are predicted structures. So we have created a common API specification that allows you access to both the predicted and experimental models and the associated annotations. And the important thing there is to also work with this community to make sure we provide the quality assessment for the structured data. But once we have access to these models, it's really just 3D points in space. And what we need to do is actually add on it some biological annotations so that these 3D models become meaningful. So we understand that there is a heme, it's interacting with histidine, etc. And so coordinate themselves are not sufficient. you need to have additional data to make sense of it. And this is even more important because most of the users PDB has are non-structural biologists. They are not experts in structural biology. There are many data resources that take PDB data and provide it to their users who are, again, not experts in structural biology. And so what we need to do is to create a mechanism and create infrastructure where we can answer questions for non-experts. And they may be as simple as what structural data is available for a given protein, and how can you download it? And this question was actually asked in one of the previous presentations. If there are multiple models, how do you choose one of them? Or are there ligands that bind the protein? Are they cofactors, drug molecules? Where are they binding, etc.? And so what we need is, alongside the PDB information, we need to enrich this data. And that enrichment can happen at different levels. So we could have enrichment at the chemical entity level, which means you can say a molecule that is observed in the PDB is actually a cofactor or a substrate or a drug molecule, to that there is a complex or an assembly of multiple proteins that is functional in a biological system. And so we need to collate all these different annotations which live at different levels and then link that back to the structural data. So at the moment, we have different websites, and this was shown in different presentations over the last 2 days. And what that also means is every one of us has some expertise and have devoted time to enriching data at each of the wwPDB sites. And that means that some of this data is available on one site and not another. And so the data is fragmented and the annotations are fragmented. We have slightly different ways in which we show that data. So there is also lack of standards for 1D, 2D, or 3D representation of both the structured data and the associated annotations. And so as wwPDB, we coming together to see how we can address these problems. And one such problem is just linking the structured data to sequence information. And so at PDBE, we work closely with Uniprot, being at EBI. We are in the same building, and we map PDB to Uniprot. And so we now are sharing these efforts with our wwPDB partners. And so, for example, PDBJ has implemented an RDF representation of this data. And so we are making this data available in different formats and through different websites, and it's consistent, and also it brings in efficiency. Similarly, as you saw, we are trying to standardize the representations as well. Visualization is quite important factor in conveying the information. And so RCSB, PDB, and PDBE are collaborating on the small star project. So we have one way of displaying all the 3D information. But one of the limitations of archive, which is static, is that it is annotated at the time of deposition. So when we get in new deposition, we annotate it, and then we release that data, and then we don't update that information. And so we need to think about how do we make sure that our users get up-to-date information. And also, given that we have archive that is static and each entry represents one particular experiment, sometimes it becomes difficult to answer scientific queries that require you to bring together multiple experiments. So for example, this is just one example of fragment screening experiment that was done on the COVID protease. And as you can see, there are so many different fragments individually in different PDB entries. To really understand where there might be a binding site, you need to bring all of them together. Similarly, you might want to understand if there are multiple conformational states. And again, you need to bring together all this information in one place from different experiments to actually answer some scientific queries. And so this is the origin of the next gen PDB. So there are certain things we want to achieve with having this next generation of PDB archive. So first and foremost, we want to make sure that we enrich the data with biological and medical context. And so we will bring together the different annotations that different sites have in one place. And that means we will have up-to-date crosslinks to different data resources, and this will be kept up to date with weekly release. And we also want to standardize some of the visualizations. And one of the other major, I think, effort in this is how do we represent a group of entries and provide access to our users to different experiments in a way that they can then derive some biological knowledge from it. So for example, if you have time resolved data, the different steps in the reaction, for example, might be in different PDB entries. And we want to make sure that we provide access to our users in a way that they see all of it together. So one of the first examples of this is to add SIFT's data into this next-gen archive and create the necessary data standards for it. So we've worked together, RCSB and PDBE, to create the necessary categories in MMSIF to represent this SIFT's data. That includes the per residue mapping, the mapping to other data resources. And this, to begin with, will be available from PDB FTP area until we create this common next generation archive that will be then common to all the wwPDB site, and it will be the single next generation archive of PDB. Similarly, we are working on creating the necessary visualization infrastructure. So as I said, we already started with MolStar, but we want to now extend it to not just display the atomic coordinates, but also to multi-scale structures and to handle multiple entries, to show superpositions, to annotate the structures for different states, for example, the conformational states that are relevant for a function of a particular complex or a protein. And we want to go further than just having a standard for 3D display and work on creating some standards for displaying the 2D topologies or the 1D sequence data and the associated annotations. So the plan in working on the visualization part is to really create a comprehensive framework to display not just the 3D information, but the sequence information, and link those together with the functional and structural annotations that the users need to make sense of this data. So the take-home message from my presentation here, and then I think we will go into a roundtable discussion, is first and foremost, I think open access is extremely important, and open access actually accelerates innovation. And we have seen that with the new the recent deep learning methods that provide predictions for structure. So to continue this and to make sure that all the associated and relevant data is archived properly and made available to the community, wwPDB is taking lead in establishing this structured data ecosystem that I described. The availability of predicted structures will certainly accelerate structural biology research. I think it's a fantastic new tool that has come along and will be used very effectively, as we have seen already from various publications that have mentioned use of predicted structures. Data enrichment and visualization are a critical part of providing this data and exploiting the structured data to understand biology. And so again, we are looking at, as wwPDB, how we work in that space to create the necessary standards. As I described, we are going to establish the next Gen PDB archive to provide up-to-date value-added annotations, and that hopefully provides the necessary biological context to our users. And at the same time, we are going to start working next year on working with the visualization community to establish standards for multi-scale visualization and all the structured data as well as the annotations. So with that, I want to acknowledge the PDBE team. I want to acknowledge the wwPDB partners. And thank you again to Chris for organizing this, and thank you to our colleagues at RCSB. And I'll stop there and take questions, or we can directly go into the roundtable.
Thank you very much, Samir. That was really interesting and thought-provoking presentation. I think it's probably better if we just go straight into the round table, actually.
Yeah. That's fine.
So I know that you and Stephen were the main participants, but we have a number of other panelists there. So I would encourage them all to contribute if they feel able to. Okay, so if anybody has any questions, if you can put them into the Q&A box, and we can then put them to the panelists. Perhaps I can start off. One thing I constantly have to remind myself is not to get seduced by these really nice images that are produced well, by Molstar and others, and remember that proteins are not static and that they do move and they do flex. How would you imagine you might be able to capture that sort of information?
So that's interesting question, and there are a couple of points there. So I think one of the ways we already have captured it is if we start looking at multiple structures together rather than just looking at individual snapshots. So that's one way of doing it. But you have to also now start thinking about having data from other experimental types like the small angle scattering, and how we can take advantage of this predicted structure, small angle scattering data, and the models we have there to understand dynamics in that context. The other, I think, major issue with the current visualizations is that we don't really represent this uncertainty in any way, except the B factors we show as colors, but that's not the same as actually representing the uncertainty or the dynamics, depending on what we are looking at. So one of the things we definitely want to address when we are working on this visualization project is to think about how we represent dynamics and how we make sure that we are also able to at the same time look at uncertainties as well. I don't know, Stephen, you want to add something?
Yeah, thanks, Samir. I concur with the comments you just made, and I want to amplify a few points about visualization, and then talk about the challenge that intrinsically disordered proteins represent. So we both using Molstar, both PDBE and RCSB PDB, can reflect the fact that many of the segments of polypeptide chains for which there are computed structure models coming from either AlphaFold or Rosetta Fold are actually not well predicted by the methods. A recent analysis by Rohit Papu suggests that that identification in either Rosetta Fold or AlphaFold of a low confidence or very low confidence region is a very good indicator that that part of the polypeptide chain does not have a well-defined 3D structure in every context. So many of the human proteins, which look like beads on a string, actually resemble spaghetti and meatballs in the computed structure model predictions. You've got well-organized domains, that many of which are actually already represented in the PDB. And then you've got parts of the polypeptide chain that are identified as low or very low confidence. And these segments of the protein are not reliable in terms of the predicted atomic coordinates. They should not be taken at face value. What Rohit's work indicates is that both Rosetta Fold and AlphaFold are probably better predictors of disorder than of intrinsic disorder than any of the existing methods that predated the emergence of these deep learning tools. So I think we're going to see a lot of emphasis in the NMR community on studying these in either intrinsically disordered segments of proteins, and that information is going to end up in the Biological Magnetic Resonance Bank, which is one of the 3 wwPDB core archives that are jointly managed by the wwPDB partners. And I think we're also going to see even more interesting use of molecular dynamic simulations, recognizing that there's biology in these intrinsically disordered segments, either because they adopt folded structures when they meet their partners. There are many examples of the PDB of such cases, or they coalesce with other disordered proteins and form these phase separated assemblies or agglomerations, perhaps is a better word, of proteins inside cells. And these phase separations and the agglomeration of these proteins together appears, at least in some cases, to be playing an important regulatory role in biology. So I think you're absolutely right, Chris, to be highlighting the importance of disorder, and to ensure that we as a resource at wwPDB are looking beyond the static structures, as you say.
Thank you. Another class of proteins which I've worked on a lot in the past are G-protein linked receptors. And one of the things there that is important, of course, is the membrane as well. And, quite often we tend to forget about that as a structural component. And I just wondered whether that is likely to become, a part of the structures in the future.
So that's very interesting, Chris, because the science paper exactly highlights this. So the nuclear pore, when they looked at it in context, so from tomograms, and when they didn't have that data, it shows a very different mechanism, or it provides you with a mechanism to see how it's controlled, the pore size, which also gives some indication of how it might control the function in terms of its activity. So this is one of the things I think that we will see going forward, that the what's called in situ structural biology, or the visual proteomics, as it was called recently, will become a way of getting structure data. And I think it's really exciting that we are going to see these structures, not just molecular level, but in their context in cells. And that really would provide us with a completely different understanding, probably, of how these work.
Yeah, thanks, Samir. I also wanted to highlight the fact that some of the cryo-EM, well, some of the cryo-EM structures and some of the X-ray structures of integral membrane proteins, GPCRs, etc., do include ordered electron density or electric Coulomb potential map density for the lipid components that are immediately adjacent to the protein. And so there are already some insights there into interactions between proteins and lipids, and we saw this, as far back as the reaction center when that structure was deposited to the PDB in the 1980s, I believe. A word of caution about predicted structure models for integral membrane proteins. It has been brought to my attention that many, if not all, of the monotypic membrane proteins with a single alpha helical span going through the lipid bilayer, when predicted as full-length proteins by the deep learning methods, end up with the hydrophobic alpha helix that would be, that sits in the membrane, actually being approximated to the surface of the globular regions that extend either extra or intracellularly, and that those predicted structure models are not reliable as a consequence. Because the algorithm didn't take into account the fact that there are lipid interactions between with the hydrophobic alpha helical segment that extends through the membrane that ensure the integrity of that part of the structure. There aren't a lot of examples in the PDB, so I don't think that the deep learning algorithm has begun to contemplate interactions between the protein, between protein and ligand. Similarly, there are potential issues with understanding how ligands bind. It's the same problem. The training set for the Greg Petzko used to say that which makes you can also break you. So the training set, the PDB made deep learning, as Samir commented, made the deep learning applications to de novo protein structure prediction possible, and contributed enormously to its success. But it also they also constitute a source of potential limitations for the methods as they're currently being implemented and utilized.
Yeah. Just this is exactly why I think it's really important to realize this is just another tool for further validation that needs to be done experimentally, and also I think when you look at these structures, you need to really understand how to see them, how to identify issues, and where these structures might be really high confidence, where they are not, and so how to use them. So some of that we covered. So we had a similar problem, I think, with the PDB, and maybe we still have. Some people think that every structure in the PDB is perfect. And I think we showed in the validation that that's not the case. And it's same for these predicted structures as well. They are predictions. And so they should be taken as predictions when you're using them.
Yeah.
I think part of the problem is that it's described as AlphaFold, which implies that it's an algorithm that predicts folding, whereas it actually doesn't.
It doesn't at all. It has
Yeah.
nothing to do with folding. It's really a prediction of structure, and in a way it's a fantastic mimic of what is observed in the PDB in terms of packing. So it's not even, in my opinion, just necessarily final structure. It's doing it locally in terms of packing, and that's what it has learned from the PDB. So just yesterday I saw the tweet about having the sequences inverted. So you take sequence alignment, you instead of going N to C, you feed it in as C to N, and it predicts a structure which is kind of mirror of what it would see if it was N to C. So it's really about, so it's a nice experiment. There are already where people have scrambled the alignments to understand how it uses co-evolution or what's the limitations in terms of the structure prediction. So there is a lot to learn still to understand how it works and what the limitations are. But yeah, it is a prediction, and that needs to be emphasized.
Yeah.
Really, every time we use the word AlphaFold or Rosetta fold.
Yeah, and let me just add one more point. That's a plug for the work that Worldwide Protein Data Bank is undertaking with respect to validation. Samir and others have correctly pointed out that the atomic coordinates that are stored in the protein data bank are in fact also models. They are models that are based on a combination of experimental data and computing, and you could see from John Berrisford's talk that there have been some instances of wishful thinking on the part of PDB depositors. But nonetheless, they're all models, and these and every model needs to have, at every position, every model needs to have some form of confidence assessment associated with it, as we see with AlphaFold and Rosetta fold. What we don't have right now is a global system for validation of structure models, both experimental and computed structure models, coming either from template methods, homology modeling, or from these deep learning methods. And it's our objective, and our colleague in the wwPDB, Jeff Hoke, who is the head of the Biomag Res Bank, which is headquartered at the University of Connecticut. He has begun with us organizing a series of workshops aimed at working with the community to develop a single standard for validation of 3D structure models, independent of where they come from. And the working hypothesis at the moment is that with recourse to Bayes' theorem, we will be able to make some headway in terms of standardizing validation of structure models between experiment and these different types of computed structure models that have become all the rage since the last in the last months. It was only a relatively short time ago that DeepMind released the AlphaFold code for non-commercial use, and working with EMBL EBI released the contents of AlphaFold DB for 21 proteomes or so, soon to grow to hundreds of millions, to I think more than 100 million structures when they release their calculations for the Uniprot 90 set that will be coming fairly soon.
Yeah. Another question. You talked about this universal database. Will you be able to add things like calculated data, say electrostatics and things like this, which would be, I think, useful to have associated with protein structures?
Yeah, well, that's just another kind of annotation, isn't it, Chris? So I think the answer is that in principle, yes. I think the challenge is going to be the richness of such potential annotations, and the importance of vetting them in some way. If there are 4 different algorithms for calculating protein electrostatics, it's historically the Protein Data Bank has not played favorites, has not tried to choose winners. So that puts us in a position, say, do we need to, would we need to integrate annotations from multiple methods for calculating electrostatics? Would we take a consensus? These are issues that are going to have to be resolved going forward.
So I just want to say, in that context, the important thing we are trying to achieve here is to begin with actually defining the data standards. So as Stephen said, if there are 4 methods and they are all putting out their results in 4 different ways, that just makes it that much more difficult to start even comparing them, and are they compared against the same benchmark, etc. So by floating this idea and trying to work with it, we are going to first kind of bring out that data standards are important. And secondly, once we bring together these different annotations, we can start asking these kind of questions about even benchmarking data sets. Can we have common benchmarking data sets? Otherwise, every publication we see with a annotation is always best compared to every other method.
I think you're absolutely right. Yeah. But you're going to have the same problem with, you have alpha fold structures, but people are going to say, well, why can't we have the Rosetta fold and yet another algorithm in the future?
But this is the thing, that was the thinking behind having these 3D beacons, because then at least all of them now can have the same way of accessing data. And that's a start. And one of the conditions in that is you have to also provide quality metric. Now, that doesn't mean that the quality metric is same, but at least we start with that. And once we bring people together, everybody asks these questions that you are asking. And then it becomes easier to address them, because then everybody has the same question.
I think that's, I think it's a fantastic initiative. I think I see it as an enormous challenge, and I don't envy you, but I'm glad you're doing it. Okay.
Something was brought to my attention in the chat that we didn't touch on, which I'd like to mention, and that concerns XFEL as a way of getting access to experimentally to protein dynamics in addition to NMR. And I should have mentioned that, and I apologize to any XFEL practitioners who are on the call. And in some cases in EM, of course, there's some heterogeneity of the conformations that one sees in assemblies, and that provides information, at least about static disorder. It may not tell us about dynamic disorder.
Yeah, this is true.
So my thanks to the chat messenger.
Well, we're coming
so
up to
Samir,
the hour.
should we thank the Royal Society of Chemistry?
Yes. I was going to say we have a minute left, and I think Chris, really thank you very much for organizing this, and thank you to Royal Society of Chemistry for giving us the chance to celebrate the 50 years of PDB in this way.
Thank you, and our thanks to all the participants. And there were at any given time more than 50 on both of these Zoom calls, which is gratifying. And I know these the talks have been recorded, and they're going to be made available by the Royal Society of Chemistry, right, Chris?
That's correct. So I'll edit them over the weekend, and they'll be up on YouTube next week.
Wonderful. Thank you very much. Yeah, it's a pleasure working with you. I hope we can do this again.
I think so. I mean, the discussion about AlphaFold and, you know, model structures and things like this, I think Samir suggested that we come back to this, and I actually think it's a really good idea, because so many people are they're so accessible now, and so many people are using them, that actually some kind of discussion about the validity of those structures is really quite timely, I think.
It has become a nice problem to have, that there are too many structures available now.
Yes.
That's right. It's the same problem as what happened with the genomic revolution.
Exactly.
I remember our colleagues in the pharmaceutical industry were lamenting the fact that there were so many different proteins that they could view as potential drug targets.
That's right.
And now the problem's been compounded.
Yes, you're absolutely right. Yeah.
As Samir said, a great problem to have.
Yeah.
Well, once again, thanks to everybody from your side who've presented and helped get everything ready, and also who've been instrumental in creating the PDB over the last 50 years. It's a resource that is, as I keep saying, is absolutely invaluable, and I hope to see much more from you guys in the future. So once again, thank you very much for your time, and everybody keep safe, and as I say, the videos will be up next week.
Thank you very much. Thanks, Samir. Thanks, PDB.
Thank you.
And thanks to the
Bye.
RCSB team.
Thank you, Stephen.
Thank you.
Bye.
Bye.